Log on, tune in, find out. Another good idea from Cambridge.
Modeling the evolutionary dynamics of CRISPR spacers. I'll leave it you with a few minutes left.
Yeah, thank you. You can hear me? Yeah. Okay. Yeah, I'm going to talk about evolutionary dynamics of CRISPR spacers. Of course, I first start to explain you what this CRISPR is about. The abbreviation stands for clustered regularly interspaced short palindromic repeats, and it really doesn't tell you much. It's a genomic structure in bacteria. So this is a part of the bacterial genome, and it has some. It contains some Cas proteins, and then these repeats, which are in dark blue, and some spacer elements of different sequence that are in light blue. And each of these repeat and spacer elements are in about 30 base pairs long. And they originate from invading DNA material from plasmids or viruses, which is here in this cartoon. If a virus is inside the cell, then the machinery detects a short motif called PAM and then integrates this protospacer into its genome. And if it does this, it can defend the virus containing the corresponding sequence later on. So this whole repeat spacer array is transcribed into a long RNA, and then there are different CRISPR types. This is not important here, just there are different proteins or even function RNAs that cut these repeats, and then the spacers like base pair with the invading DNA, and then the invading DNA is degraded. So what we need for this talk later is that if a virus infects the cell does have a spacer that exactly matches the genome. It can defend the virus and live on. But if it doesn't, the virus can multiply inside the cell. Of course, given that the virus also overcomes the other defense mechanism inside the bacteria, the phage proliferates and destroys the bacteria. So therefore you see that there's a strong pressure towards integrating matching spaces if a virus is around. And so we know that the sequence of these CRISPR arrays tells you something about the virus, bacteria dynamics going on. And we want to model the insertion and deletion rates of these CRISPR spacers in a phylogenetic framework. Yeah, just to give you an idea how data looks. So the numbers are different spacers, and I colored the numbers if they occur multiple times, and the lines are different strains. We sequenced Staphylococcus pseudo intermedius, which is of CRISPR type 2. And you see that, for example, some strains share whole stretches of spacers, and some have largely unique spacers. So the idea is that we just code the spacers binary as absence and presence, and we assume that they change along the phylogeny. So this is just a normal binary model. So they change according to a continuous time Markov process with insertion rate alpha and deletion rate beta, which has the well-known instantaneous rate matrix and stationary distributions. However, I told you in the biological introduction that it may be useful to have one spacers at one time point if the virus is around. If it's not around, then these spacers get lost again, So we assume that the usefulness of a spacer is also changing over time. So the minus state indicates that a particular spacer sequence is not beneficial, and plus indicates that it is beneficial because you're likely to meet the virus soon. If you put these parts together, you have a insertion rate in the minus state, which is probably low because you don't need the spaces, then you insert it. And you have an insertion rate in the plus state, and these insertion rates are different. And you switch between these two models with rates R and S. So this is a switching model or Markov modulated Markov chain, and similar to the switching models which were used to in the covariant models that are maybe familiar to some of you. So then if you put this all together, the Q matrix looks like this. And so now we have the state 0 minus and 0 plus. Of course our alignments are not labeled like this, so zeros in the alignment are ambiguous states that represent 0 minus or 0 plus, and analogously for ones. So to see whether this model works at all, we first set up some simulations. So we randomly generated Ewell trees of 100 taxa with branch lengths drawn from the uniform between 0 and 0.5. It generated binary alignment of 300 sites, and we deleted only 0 columns from these alignments because we cannot, if a spacer is not present in any of the bacterial strains, we don't observe it. And then in the likelihood function we correct for this ascertainment bias. So we assume that these spacers evolve along this species phylogeny, so we take the true tree as given for now and estimate the parameters along the along this topology. The simulation I set up looks like this. Because there were 6 parameters to set, so I simplified it a bit. I set the insertion rate for plus state to a constant C, and the insertion rate in the minus state for 1 over C, and both deletion rates to 1. And then also the switching rates vary. They are equal, or it's more likely to be in the plus state, or that it's more likely to be in the minus state. The simulation results, I summarized the deviation, which is given here just the absolute deviation relative to the true value for these 6 parameters, and also for these ratios, which give you an idea whether this process is captured in stationarity. Then over 100 simulations, this looks like this. So the rates are well estimated, but the switching rates, they deviate from the true value, and there are even some of these values which lie outside the plot. And so this is for C equals 2. So for insertion rate and plus state of 2 and insertion rate in the minus state of 1/2. So they are still quite similar. If you increase the picture gets more more messy. So the deviations increase, especially for the switching rates. And if you go to C equals 10, and also the ratio of the rates is not well estimated anymore. So I summarized the simulation in this heat plot, which just shows the median of this deviation I showed in the box plots before on a log scale, which you can see here. So for these 3 are the pictures I showed you already, and then for C equals 1, it's actually a two-state process because all the weights are the same, and of course you cannot estimate S and R because both Q matrices are the same. If there's if the switching rates are set up such that you are more likely longer in the plus state, the deviations are lower, probably because this process is quicker and we can see more mutations in the simulations. And on the other hand, if we spend longer in the minor state, which is very slow, like the insertion rate is just, for example, 1/10, the estimation doesn't work well. And one other thing to mention is that also if we cannot, in these settings where we cannot estimate the switching rates well, the ratio still works. Yeah, so this let us hope that for reasonable parameter values we may be able to estimate these parameters. And the next question was whether we can distinguish it from the two-state model. So this is the same simulation I showed you for equal switching rates, and pi 0 I just take the sum of pi 0 minus and pi 0 plus, and then if I estimate the parameters under the switching model, this pi 0 about matches the expectation under the of the stationary distribution under the two parameter values. However, if I if I estimate the parameters under the two-state model, we see that the pi 0, which is then just this ratio, is biased and does not does not match the two values. So we see that the switching model can also be distinguished from the two-state model, and now we are ready to apply it to our data, which I showed you in the beginning of Staphylococcus. It's a bacteria which infects dogs, which is maybe not so exciting, but this is the sequencing that was done in our lab. So we know from one reference genome that the structure looks like this. There are some Cas proteins, and then there is this structure of repeats and spaces, and we know the flanking genes which we use to amplify these sequences in more strains. So far the sequences are finished for 16 strains, which gave result to 162 different spaces, of which 117 are unique. So about 50 of them occur in multiple strains. And a blast search gave us the result that about 35 of them we know from which external source they came from. So there was a reference phylogeny published for these strains of 4 genes, and if we put the spacer data I showed you in the beginning on this phylogeny, you can already see some problems. So here was what I showed you in the beginning, where you have consecutive shared spacers, which is nice, and they also occur together in the species phylogeny. However, here you see that between these two strains there are also a lot of shared elements, and they are on completely different places in the tree. However, we use this tree first to estimate the parameters, and the result is not very satisfying, because if we estimate the branch length, most of the internal branches collapse to 0, and it's not really a phylogenetic model anymore if there's not much shared information. And also another point is that the switching rate gets really high, and it doesn't tell you so much. If we estimate the parameters under the two-state model, we see that the switching model is actually preferred, using a likelihood ratio test. However, as I told you, it's not very, so the spacers don't really cluster in the tree. So I wanted to have an idea first whether using the species phylogeny is somehow influencing the parameter estimates. So I used I computed a parsimony tree from this binary alignment, not because I particularly like parsimony, but because the workshop was approaching and I wanted to have an idea how this how the parameter estimation works on a different tree, which more reflects the space evolution and not the species evolution. What we see is that then there are some internal branches kept, and but then when we compare it to the two-state model, the likelihoods are not different anymore. So this has two implications. So first, also from other data I have not shown you, we assume that there's actually horizontal gene transfer going on between the strains that can maybe exchange these CRISPR elements. So we cannot use the species phylogeny, what we wanted to do in the beginning, and just estimate our parameters. But it makes more sense to do the parameter estimation together with a phylogeny estimation based on both models, the switching model and the two-state model, and then compare the likelihoods. So this is still ongoing. So to summarize, I presented the two-state model and switching model for spacer turnover, and I compared them in simulation. In our Staphylococcus data, the space evolution does not really, the data actually contradicts the space evolution along the species phylogeny. And if we use a parsimony tree, we don't find support for this switching model, so we need phylogeny inference to properly compare the two models. I want to thank John, which was actually on the first slide I wanted to mention in the beginning, and also Ross, who provides the strains, and Flavia, who did the lab work, and thanks for your attention.
Questions?
So, with your R and S, are they? I'm a bit worried whether they're identifiable or not together, because your ratio was coming out accurately, but your ability to estimate them individually was not. Have you tried looking at, say, a likelihood surface of the two of them and see if it's this unique optima, or if it's a line or something?
I have not. So I don't know about theoretical identifiability results that apply to this, and also I have not looked about the surface so far, but it makes sense. But then, I mean, if we just know that that the ratio works, that we can say, okay, the switching occurs more often in the direction of minus would be expected, then we already can learn something.
So I guess in your model, you model that the spacers evolve independently from each other, and I'm wondering if in this immune kind of context, wouldn't they be highly correlated? Like you need one space, if you have one spacer, you don't need 10 other ones.
Yeah, you don't need 10 other ones for the same, which target the same virus. That's true. But this information we usually don't have. We don't know these 10 are from this virus, these are from this virus. So this information you cannot get. And there's also another problem is that maybe if these CRISPR arrays get too long, then deletions are more likely. And this also we don't model at all. And something I also didn't tell you is that usually these spacers are inserted in the beginning. So we also have an order information. And so far we had no idea how to include this in the model. If there are any ideas on this, would be helpful.
No other questions? Then thank you.